Namaste students. I am Javin Dr. Aziz Mohammed Khan from Department of Food and Biotechnology, Faculty of Agriculture and Veterinary Science, Jyoti Vidyapith, Women's University, Jaipur. Today I am going to discuss about the topic chemostat. Chemostat and turbidiostat. These are the two terms which are used in the fermentation technology. And these are the two terms which are directly related to microbial growth. Okay. So, chemostat is a type of continuous culture. Okay, it is used in the continuous culture. We know what is the continuous culture. Continuous culture is a culture method in which we add the medium, we add the culture, and we take out the product at the regular time. Okay, that is the continuous culture. So, in the continuous culture, chemostat is a term which is used in. And chemostat meaning is it is a type of continuous culture in which a constant chemical environment is maintained in the process. For example, uh, this is a vessel. This is a vessel or this is a fermenter in which a particular environment is maintained. This environment is maintained for the fermentation process. So, which environment is maintained? The environment meaning is that the culture media, okay, the culture media are the environment. The nutrients are the environments. The chemical are the environment. Okay, so this is the environment which is regularly added and Uh, these nutrient media are regularly added at the regular time. Similarly, they are also taken out. Okay, they are also taken out at regular time. So in the chemostat method, what it is actually meaning is that the dilution of the nutrient exchange is maintained. Means it is constant. The dilution of a particular nutrient media and the culture media and the culture is maintained. That is the meaning of the chemostat. Means we are adding and we are taking out the constant amount of some nutrients in the culture at regular time. Okay, we are maintaining a condition in which we are maintaining a particular condition. So in this chemostat, they some sterile media is added, and the media and some culture cells are taken out at a regular time, so that a particular amount of chemicals should be maintained in the culture media. Okay. So, a chemostat proceeds with the limiting nutrient in the culture, and the limiting nutrient is one of the essential nutrient. Means at the starting of the method, we add a small amount of the nutrients, small amount of the culture media. But when the growth is observed of the microorganism, then what happens? We add stress media and we take out the old culture from the fermenter so that a particular conditions can be maintained. Similarly, we also add some microorganism. So that is the basic of the chemostat in which we maintain the particular kind of constant chemical constituent of the media. We maintain it. The repeated fed batch culture is closed culture system in a sense introduced, and the essential open culture is the chemostat. A chemostat consists of culture in which fresh media is continuously introduced, introduced at constant rate, and the culture volume is kept constant by continuous removal of culture at the same time. So two things are happening here, uh, in which the supply of single nutrient controls the growth rate. The fermenter is called chemostat because the growth rate is controlled by the availability of a single component of the media. Important features are that it is the continuous introduction of fresh media that hit the limiting substrate to the culture and the dilution rate. So this is the diagram of the chemostat in which a constant condition of substrate is made. Substrate meaning is that we are adding constant amount of chemicals, constant amount of substrate means we are adding constant amount of nutrient media and the microorganisms also. That is the concentration. That is the constant concentration. So the main feature of chemostat are volume of the culture remains constant. The time required to mix a small volume of a medium with the culture should be small, and the environmental conditions can be maintained constant, such as the pH, temperature, and antibiotic concentrations can be another factor, but environmental conditions should be maintained constant. The specific growth rate can be varied from just above a zero. The environmental conditions such as pH, temperature, oxygen tension can be varied, and biomass properties such as micromolecule composition and the functional characteristics can be maintained constant. And the biomass concentration can be set by editing the concentration of the limiting substrate and the maintained constant according to the required amount of the media. The substrate limiting growth can be maintained indefinitely, and this can offer relief of capable repression and the induction of secondary metabolism at low concentration. So this is the formula of dilution rate which determines the growth rate of the culture. D is equal to F upon V. So D is the dilution rate. Dilution means if we are in decreasing the concentration of any liquid molecule, then it is the dilution. We can we can dilute anything by decreasing the concentration of any substrate by adding water. Okay, here F is the volume flow rate of the medium and the V is the culture's volume. So this is the formula of analyzing the uh, dilution factor, and according to dilution factor we can manage the chemostat, means we can manage the condition of the culture media. So our lecture is over here. Thank you very much.